Good morning, good evening. I hope you're well where you sit and listen or stand. Today's going to be great. It's all about plant secondary metabolites. And I'm going to go into a pretty decent detail about their pathways, what they do, how to get the best out of them, and then what temperatures they are volatile at, so you'll know what's safe to use. Like if you want to get at a lower temperature to get a different secondary metabolite or a terpene profile brought out in your end result, you can do that. I'll show some charts for all those types of things, and I've got plenty of slides to go through too. So today it's going to be good. I support the law, so I have to respect the law. It's the law and I've got to respect it. So I hope you do too. I don't promote any unlawful activities or anything like that. And I only support organic growing. I don't support all of the chemicals and other additives that have been put into it, because it's all about sustainability. I study environmental science, so you want to do it for our children's children too. So that's the little intro thingo. Yeah, it's going to be good. I got a few good slides to go through too. And hang on, let's put this thing up. How do you get that one? Remove the stream. No, this one. There we go. It's all about secondary metabolites. Plant, medical cannabis, secondary metabolites, terpenes, flavonoids, what they do, why they're healthy, and how to get more out of your meds after you know it. Jono, who cares? Good morning, finally. Hope you're well where you sit. It'd be pretty dark where you sit, I reckon, mate. Thanks for joining. Thank you, everybody else, for joining too. The questions, I'll try and keep on top of them, but it might be hard because I'm going to be going through quite a lot of slides, but I'll still try. And if you've got your questions, try and put a question mark at the end there so I can see it. It'll sort of get highlighted a bit, and then I can concentrate and make sure that I do answer it. Cheers, Lene. Chat. Yes, good morning, good evening to everybody. Righto, this is going to be good. So, plant secondary metabolite. What are they about? Why are they there? Let's start and share and explain with some slides. Share screen. Righto. So plant secondary metabolism. So what is it? It's the sum of all the chemical reactions that take place in an organism called the metabolism. It's made up of a catabolism and an anabolism. And most of the carbon, nitrogen, and energy ends up in molecules required for proper functionings of the cells and organisms. Most plants divert a significant proportion of assimilated carbon and energy of the synthesis of organic molecules that have no obvious role in normal cells. They're known as the secondary metabolites. And how are they different? Well, there's a primary and a secondary metabolites. So with the primary, I'll read this first. At the biosynthetic level, primary and secondary metabolites share many of the same intermediates. They are derived from the same core metabolic pathways. Like secondary metabolites generally occur in relatively low quantities, and their production is widespread and restricted to particular families, genre, or even species. Yeah, that's the key thing. Secondary metabolites occur in relatively low quantities. So that's involved with your terpene profile and your flavonoid profile is very, very important in medical cannabis, because we want to retain all those health properties. I'll go through them later on, what they do and why you should retain them, and why you shouldn't. Your post-harvest operations should be in such a way where it should smell the same as when it's growing on the plant. I consider it a failure if when I'll smell the plant growing, and then when it's after it's cured and dried, and then I'm trying to vape it and I don't get that same result, I consider that a failure, because I've lost some of these terpenes and flavonoids. So this is the pathway for the plant that it comes in. So as it photosynthesizes, it produces its primary metabolism, which is proteins and chlorophyll, lipids and carbohydrates. And then it'll go into the secondary metabolite profile, and it'll start producing terpenoids, alkaloids, and cyanogenic glycerides. And then after that, it goes with the phenolics. That starts to break down a bit and just see what pathways are needed on the plant to see what it signals. And it can either go into phenolic acid, which ends up producing lignin, or it can go into coumarins. And then after the phenolics, it'll get processed again, and it'll go down to the chalcones, which will produce your flavons and your flavinoids down here, and it goes right down to flavinols, and that's when you can get your anthocyanins. So right at the end result. So that's why when to bring out your anthocyanins, it's a temperature thing that I've done through my studies, and I've got a good video out on that. And that's the end result of the pathways, as you can see here too. And that's to bring out the real purpley, the dark, bring out the blues in your genetics. It's very important actually, because it's another health property. These are all health properties. So the more healthy you want it, just try and retain and do the correct pathways to get that. So how are they different? Yes, so that's what, it's just they're relatively low quantities. So to maintain all of their good properties, you want the correct procedures. I've already said that. Their role, secondary metabolites have an important ecological function in plants. So why are they there? So if you can understand why they're there, you can manipulate them to bring out different profiles. Like I've done it out of, what's an example? Pink Death Bubba. I've had 3 solid different profiles out of the same mother plant. That's by only manipulating their secondary metabolite pathways. And this goes into quite good detail on how the plant makes that happen. Like, why does it happen? What genes, with 35,000 plant genes, are initiated? What pathways does it take? How can you alter them? Can you use any precursors or any? I'll get into it. So secondary metabolites have important ecological function in plants. They protect plants against being eaten by herbivores and against being infected by microbial pathogens. They serve as attractants as well for odor, taste, and color, for pollinators and dispersing animals. They function as agents of plant-to-plant competition and plant-to-microbe symbiosis. They increase the reproductive fitness of plants by warding off fungi, bacteria, and herbivores that maybe also make them undesirable for humans. The ability of plants to compete and survive is therefore profoundly affected by ecological functions of their secondary metabolites. So cool. And there's 3 different sections of plant secondary metabolites, and it's terpenes, phenolics, and nitrogen-containing compounds. So I'm going to touch on the first 2, and specifically terpenes. Phenolics is when it breaks down, like you saw before, and goes into the flavons. So I'll go a little bit into the flavons and flavonoids, because they're also an important health factor that you want retained as well. Is that what I was up to? Yep. So that was what I went through before, showing the chart. So I'm just going to touch. At least we're up to here now. We've progressed. Oh, there's a couple of things in chat. I'll just say, g'day. How you going in chat? Had to scream. Jono. Oh, yeah, harvesting tomorrow. It's a good time for harvesting. You can retain. Make sure you do your post-harvest operations correctly, drying and stuff, hang drying in good humidity, temperature out of the sun, and you should be able to retain them. Ned Kelly, good morning, Ned Kelly. How are you? Chimu, Sema, good morning. How's it going? It's all about plant secondary metabolites. It's going to go into good detail on how, yeah, you'll see. Get back to it. Share screen. Up to here. So terpenes. What are terpenes? The terpenes, or terpenoids, constitute the largest class of secondary metabolites. They're very diverse and and include substances that are insoluble in water, so they float. So if you use people who extract, it's very important that you want to have a terpene profile before you're going to extract, because a lot of them are insoluble, meaning they'll float, so therefore you can somehow extract them. Certain terpenes have well-characterized functions in plant growth development, and so can be considered primary rather than secondary metabolites. Gibberellins and brassinosteroids are very good. The vast majority of terpenes, however, are secondary metabolites, presumed to be involved in plant defenses. See that? Defenses. So terpenes are toxins and feeding deterrents to many herbaceous insects and mammals, microbes. Monoterpene esters, called pyrethroids, found in the leaves of chrysanthemum species, show striking insecticidal activity properties. In conifers, I'm not going to read the conifers because this is to do with medical cannabis, but pyrethroids are very good a botanical organic approach to controlling insects. And they're a monoterpene, as it says. Many plants contain mixtures of volatile monoterpenes and sesquiterpenes called essential oils that lead to characteristics odors like peppermint, lemon, basil, all these different properties that we get out of our medical cannabis. There's hundreds of different ones in those different varieties. That's why it's such a medicinal plant. They are frequently found in glandular hairs that project outwards from the epidermis. They're also stored in plastid aleoblasts. Hang on, I've got a chart here. These are the different plastids that are in the plants, and you can see here the storage of lipids and oils in an aleoplast. So you all know chlorophyll, which gets stored in a chloroplast. So these are just other plastids. This is a common one, a starch, the amyloplast, which is the storage of sugars and starch itself. But yeah, just to touch, that's where, and the other places where the plant will store different elements and molecules. I've got to just touch on these. Triterpenes defend plant against vertebrate herbivores. Include saponins. Saponins are steroid-like triglycerides. Phenols. Remember, that's another section of it being broken down. So I'll just show back here again. The phenolics. See coming down, so that's the terpenes, and this is into the phenolics now. So phenols, plants produce a large variety of secondary compounds that are called the phenol group, a hydroxyl function group of an aromatic ring. The substances are called phenolic compounds. Plant phenolic compounds is more than 10,000 of them. There's heaps. Some are only in organic solvents. Some are water soluble, like carbolic acids and glycerides, and some larger insoluble. So phenolics play a variety of roles in plants. Also, it's to do with defense against herbivores and pathogens. And they also attract pollinators and fruit dispensers. No, not really in medical cannabis. It's more to do with defense. They don't really want to attract a lot of things in reducing the growth. Yeah, phenolic compounds are also involved in absorbing harmful UV radiation. See that too? So that's why greenhouses mightn't be as potent as ones that are grown outdoors, because of that secondary metabolite pathway hasn't been activated in reducing the growth of nearby competing plants. Okay. Phenolic compounds are of two principal types, the carotenoids and flavinoids. So the carotenoids, they're the yellow orangey bits that you see in the leaves that when it ages, the leaves it starts off with chlorophyll, and once that pigment's used up, it'll drop it and it'll go into the yellowy section, and that's with the carotenoids. And the flavonoids, they include a wide range of coloured substances. So they're mostly grouped with like the purples with the anthocyanins, And then it's also, when you're bringing out your other ones, your reds and your pinks and your blues. And then there's two types of other groups of the flavonoids that are also found in medical cannabis. You have flavons and flavonols. So flavons, O-N-E-S, and flavon, O-N-O-L-S. The other two types. And they would absorb light like UV rays, and they're also good against warding off against some insects. Phenolic compounds itself, don't think I need to get into that much. Compounds, or a release of secondary compounds by one plant that has an effect on neighboring plants is referred to as allelopathy. This is how plants speak to each other, through this phenolic compound. So they'll go and produce it. It'll go through this pathway. It'll go in here and produce it. And then once it comes in there, it'll go into a methyl pathway where it gets broken down again and it goes into, have I got any of those? No. Those real detailed pathways where it starts to change into a methyl, which means it's going to go volatile, and it goes into the air, travels, and another plant's receptor picks that up, and then it changes its methyl back to the normal signal, and it picks it up, and that process is called allelopathy. If a plant can reduce the growth of nearby plants by releasing chemicals into the soil as well, it may increase its access to light, water, nutrients, and its evolutionary fitness. Because plants, they're always looking to their best, get the best out of themselves, and to reproduce if they're under too much stress, because they want to continue. Alkaloids. There's a lot of alkaloids too in the plant. I'm not going to talk about them. Here's a pathway. What's this one for? Photosynthesis. It comes in and it makes these primary carbon metal. So in the pyruvate, that's just, okay, this is energy. Oh, there was terpenes down the bottom. So there's a very quick pathway to show how it gets into the terpene profile. It's a bit detailed, so I'm not going to go into it. You're probably not really interested. Plant defenses against insect herbivores. So plants have developed a wide variety of defense strategies against insect herbivores. These strategies, there's two types, constitutive and induced. So the constitutive defenses, they've got, they may be stored compounds in the plants or precursors of active compounds that can easily be triggered. So that's why if you've got you prime your plant, it's called, with breeding, and you put a genetic resistance into it, it does this, it activates this pathway by having your genetic genes already primed. And I've done that to my genetics. So they're all just sitting there waiting to their defenses just to get ready to go. Most of the defense secondary compounds are constitutive defenses, not induced. So the induced ones, they're initiated only after an actual damage occurs. So once if you bend it, if you crack it, you put it into the UV, if your insects and pathogens or anything bites it, gets into it, starts messing with it, that's an induced defense. And that produces defense proteins such as lectins and protease inhibitors, as well as toxic secondary metabolites. So in principle, induced defenses require a smaller investment of plant resources than constitutive defenses, but they must be activated quickly to be effective. It's so cool. All right, plants can recognize insect saliva even. This is, I suppose it's a pathway of the secondary metabolites, just proving that the different types of things. I'm not going to go into that one. There's this shows other pathways too. I may just, all right, see if there's anything going on. It's in chat. I'll put that back up. Okay, cool. Oh, yeah. It's weird you were silent. Internet dropping out. No, it's pretty good. I got a good internet connection. Thanks for letting me know, though. Oh, it's fine for me. Oh, yeah, good. All good. Yeah. Thanks for the heads up, though. It looks clear on my screen. Oh, that's no good. Is your page blurred? I don't know, maybe check your internet speed, sorry. That's no good. Lucky it's recording though, so you can watch back. Right, I'll get back into it. This one. A major signaling pathway involved in most plant defenses against insect herbivores is the octadecanoid pathway. That was an exam question, which leads to the production of a plant hormone called jasmonic acid. It produces jasmonates, and jasmonic acid level rises steeply in response to insect herbivores. Jasmonic acid is synthesized from the linoleic acid, which is released from the plant's membrane lipids. That'll do. Okay. Some plant proteins inhibit herbivores' digestion. Well, this is to do with secondary metabolites. Yeah. For example, let's get on with it. I've got a lot more slides to show too. Herbivore-induced volatiles have complex ecological functions. Herbivore-induced volatiles. So the induction and release of volatiles in response to insect herbivore damage, combination of molecules emitted is often specific to each insect, and they can release different pathways from doing them. Like there's a what's that? Some antimicrobial complex before. Come on, it gets to it. Because I used to call it the FLG22 gene, but it's not. It corrected me. Infections induced by anti-pathogens. The phytoalexins. The phytoalexins are pretty important. It's a response to bacterial or fungal infections. It's a synthesis called phytoalexins. They're a chemical group within the secondary metabolites that produce strong antimicrobial activity and accumulate around the site of the infection. They're pretty common with the hypersensitive reaction in medical cannabis. When you'll see something will affect it, it might get onto a leaf, and then you'll see that the leaf start dying around in a round circle. That's because that's activated this pathway that's formed the hypersensitive reaction, that's called, and it's because it's killing all the cells around the problem to stop it spreading. As an example. Phytoalexin production appears to be the most common mechanism of resistance to pathogenic microbes in a wide range of plants, including medical cannabis. Phytoalexins often increase after pathogen attack. Yes, plants do not store any of these enzymatic machinery required for phytoalexin synthesis. After microbial invasion, they begin transcribing and translating the appropriate messenger RNA and synthesizing the enzymes for the action to take occur. Some plants recognize specific pathogen-resistant subjects. Here we go. This is cool. The microbe-associated molecular patterns. It's called MAMs. It's so cool. Oh, here's that gene. So I thought it was that one. It's wrong. It's the FLS2 gene receptor in the plant. That's the one that can detect bacteria that have the little squiggly tail at the back called the flagella. And then the PEP13 receptor in medical cannabis, that recognizes all of the Umiceti pathogens, which is your fungus down in near the roots. That's like Pythium, Fusarium, that'll do. A form of defense strategy is also referred to as innate immunity. I'm not going to get into that. The single within a pathogen. Infections. We're going to get into role. No, that'll do. So that's the thing for the first slide that's done. Now I've got the second one to go into. Any questions? Having a bit of a squeeze? No, looks pretty good so far. I'll keep going. So some roles of some flavonoids. Oh, this is an example of the genes in plants. So the expression of these genes is activated when bacteria perceive specific molecules, like we're just talking about the I've got to see it a few times to learn it. FL2, FLM2. Someone, they're saying, no, it's not this. Oh, it's FLS2. So the flagella, FL. So how can I remember the S? What? Flagella. There's nothing that, when they name these receptors and genes, they always put it like here. Look at the epitope here at the start. It's a FLG. So that's a flagellum. You can remember that easily. How can you remember the S? FLS2. I'll just have to try hard. So the expression of those bacteria, they're in general flavonoids that are secreted by roots of the plants. So there's three different, the nod factors are this gets a bit technical. There's three different ones of those, the nod genes. But you don't really want to hear about all that. That's just an example of the flavons and the flavonoids, how they interact. The stage of, no. Next one. So it went on a bit about, so all of the terpenes, they're volatile oils. Like you'll get them, lavender will have linalool, for instance. And in different cultivars of medical cannabis, you get ones with linalool in it. So singly, you can go and have lavender and get in higher amounts, or you can have it in your medical cannabis and know how to get the correct properties out of it. So after these, we'll go through these. I'll show you some temperatures on which to get the best out of it, to unlock it, because they all got a temperature in which they are unlocked at, is the best way to put it. This is, so the presence of volatile oils in plants, like they're in the flowers and the leaves. And as you know, there's heaps more in the flowers, and in those plastids before, they're also stored there. In the dermal tissue, there's different other places where they can be stored too. The presence of volatile oils in plants is seeds. We don't have much volatile oil in our seeds at all. There's just the hemp oil, which isn't, it's got properties in it, but there's no like CBD in hemp oil. Just an oil in the bark. Yep. Woods, roots. I don't think there's much in our roots, and in the rhizomes. No, we don't have that. You don't want to harvest that. Maybe the bark. I've never heard of using the bark. The stems do get used. There was a physio in BC, and he was buying our stems. Peels, berries, fruits. You know about fruits. There's the stems there. Localization of the volatile oils. So trichomes, secondary canals and ducts, secondary, it's edioblasts. Remember? Eleoblasts. E-L-A-I. E-L-A-I. Sometimes they get it wrong too. Properties of volatile oils. Well, you all know the properties of them. They're all different, volatile at different temperatures. When they're all liquid at room temperature. Oh, really? Yeah, that's true, because ocimene and CBG is volatile at 60 Celsius. It's one of the lowest ones, or 150 Fahrenheit odd. So most of them are insoluble in water. They're lighter than water, with exceptions, and soluble in organic solvents. Yes, so they extract them. Properties of volatile oils, highly refractive, okay, on long exposure to light, to lead to change colours. Biosynthesis, okay, I'm not going to get into that much more. This is the biosynthesis, you don't really want to know how they get to that. Composition of volatile oils. So this is the bit that I've sort of explained already, how there's mono, sesquiterpene, triterpenes, and it goes on to different other classifications. Actually, it's probably might be in a good time to talk about that. The different classifications of, I'll get to there. You don't understand. Volatile oils are classified on their basis of their composition. Yes, it's their isoprene level. All right, if this doesn't go into it, I will. Oil-rich ones. Okay, it doesn't go into it. So terpenes. So where's their isoprene level? here. So here is the monoterpenes, and see how it's got a number of 10 carbon atoms. It has 2 isoprene units. Then it goes down to sesquiterpenes, and then diterpenes, then sesquiterpenoids, and then triterpenoids, then tetraterpenoids, which are the carotenoids. And that's how they rate. And then underneath the different ones, you've got your different ratings by how many carbon atoms are actually in there. Very good. Back to it. Explains that. No, we'll finish that one. This one. Okay. Linalool, I was speaking about before. That's its little chemical composition. Yes, so we've got sandalwood. As you know, the sandalwood is very dominant, peppermint. All these different types of terpenes are in medical cannabis. So most of the time, they used to have to go and get one out of lavender. They just go and get that one type out, the linalool acetate out of the lavender. And now you can get it out of medical cannabis if you've got the right ones. So they're so medicinal. Lemon, yep, limonene is that terpene name. Eugenol, that's very handy. I think eugenol, it's an close, eh? All right. These have got all health properties of anti this and anti that in them, and they all vary. So each one has their own benefit. That's why people get into essential oils. So you can get them all out in medical cannabis. And the factors that affect the different types before are stresses, to cut a long story short. And that can be temperature, the number of hours of sunshine, the frequency and magnitude of rain, can be the soil type, what's in the type, what type of genetic makeup you have in the soil. Is it fungal dominant or bacterial dominant? Water stresses and drought stresses, other abiotic and biotic stresses, insect, microbial stress, locations, harvest, integral spectral rate, wild and cultured plant material, very good plant part. Okay. Well, I'll stop and see if there's any questions, because that's the end of this one. What's the next one? Oh, up to that one. Okay, we're up to the big one. Cool. Got to a section. All right, let's see if there's any cues going on. Plagesburg. Hey, Cameron, how you going, mate? Yes, I have a room that looks like... Jono, who cares, says, I have a room that looks like it gets a infection. The leaves are thin and look like when you don't give them enough water. Ah, so they're sagging. Oh, so yeah, if you've got the right amount of water, that's definitely telling you something's going on. And you can tell from leaves there, if it's got any color, or the margins, which is the edge of the leaves, if they're shaping up or down, or if there's different types of hypersensitive reactions, deaths going on inside of the leaves. So you really read them. And if you don't see anything like that, if it's just wilting down, there's a high chance that you've got a fungal problem in your substrate, mate. It'll be Pythium or one of those Umiceti pathogens, and that's what I reckon. So it's to do with the water that you get, that you put in, that has those pathogens already in there. And if you give them the favorable conditions, that's how they can repopulate themselves and take over. So you have to test, therefore, once you got it, so now you've diagnosed what it is, you have to now eliminate it and make sure it doesn't happen again. So you go, well, how did that happen? Well, it's from your water, probably. So you test your water extensively, or change your water, or get a reverse osmosis set. You want to get a 5-stage. I don't think you have one. Get a reverse osmosis, mate. It's probably what's caused all this. Thanks for the question, though, mate. Cameron, there was an old rumour about eating the root. Okay. Love them all, yum. Yes. Present. I thought I'd start a little bit later to get a few more viewers. So that's the reason why I started a little bit later. Okay. Okay, the water is in Thailand. Yeah, that's what I thought. Jono, you've got you're using Thailand water, you've got to get a filter on it, mate, because that's how you've got your Umiceti pathogens. So what you can do now, you have to get a lot of, get some Bacillus pumilus. That works really well with Pythium. And what's the other one? Some good biologicals that are going to really help. And you want to keep it a bit dry. There's a chance that you're just going to have to start again. So I'd clone if it is and fruit them out and start again. It's probably your best fit. Hey, Teo, how you going? American one. This is a good one on secondary metabolites, mate, just on how they work. Probably gone into it a bit more about the plant defenses and how they form inside the plant and what the plant does. I'll get back to it. I'm pretty much at the end, though. Yeah, good stuff. Where is it? Share screen. So this is going to get into all of the temperatures in which they're all volatile at. Is this one? No. It's this one. So this is how the terpenes, it gives their classification through the isoprene units. And then under that, it just breaks down, goes into different, what the classifications are. So under monoterpenes, they have 2 isoprene units and 10 carbon atoms, as you can see there. That's how they're all rated. And the difference is they're classified, their classification's all different, and they're makes up of all different things. They break down differently, and their molecular structure is all different too. Dronol, it's rad. So how can I summarize these? Because it goes into a heavy detail, and I don't want to go into that much detail because it'll be very confusing in different types. I'll go into the sesquiterpenes. That was the next one on the chart. So I should give examples, actually. Some good examples in medical cannabis of the monoterpenes are myrcene. Myrcene is very good for pain. And then you've got your ocimene. That's another good one of the monoterpenes. You get 60 degrees Celsius or 150 Fahrenheit, and that starts to volatile off. You've got your linalool and drolinol. They're good monoterpenes. That'll do. And some really good sesquiterpene terpenes, some examples of them in medical cannabis that are common. Humulene and humulene is common. Not going to make any up. Bisabidol, that's common too, because I like saying that one. Bisabidol, the bisabol, bisabolane. It's bisabidol is the terpene, but bisabolane derives. It has a 6. Oh, actually, I'm not going to go into that much detail. All right, and next one. As an antagonist, okay. The sesquiterpenes, it's a tricyclic. Here we go, diterpenes. So they have 4. So what's some of them? Phytol, that's a pretty good one. That's a real good one, actually. And the sesterterpenes, is there many of them? They're getting up a bit high. Tetras. The carotenoids. All right, it's going up a bit. See, this is all the uses of them, of the terpenes. They've got anti this and anti that. Anti-cancer, antibacterial, antiviral, anti-inflammatory, antioxidant, antispasmodic, anti-parasitic, all sorts of antis, just you can think of them. That's why people use volatile oils. Anti-cancer terpenes. So, is there many people watching? There's not many people. So, yeah, I'm not going to actually talk about that. Antiviral terpenes. Anti-inflammatory terpenes. Here's a pathway of the monoterpene pathway that goes from different plastids. So I'll start off in the leucoplast, which remember the leucoplast. It's the synthesis storage place for fatty acids and amino acids. and that type of plastids in the plant. It starts off there and goes into the organelles inside of the plant, into its endoplasmic reticulum, then into the mitochondria, and into the cytosol, and then it's available. What's this one? Oh, here's the Wikipedia terminology of a terpene. Terpenes are a class of natural products consisting of compounds with the formula C5H8, comprising more than 30,000 compounds. So cannabis, medical cannabis, would at least have 10,000. So you've got all those different ones, go through all those different isoprene levels, and are all available in cannabis. So that's why you've got to see which ones under these different terpenoids agree with your body. So it's all good and saying, yeah, I just want some more. But you might find that myrcene is really, really beneficial in a high myrcene dominant, or beta-caryophyllene is very good for pain too, those types of ones. All right, that'll do for that. Let's get back to the, stop sharing. So I'll get back to some temperature stuff. Temperature. Here we go. Anything said? Hey, what are you up to? Hey, Kelly, Kemet, Gorkowski. Hey, Gorkowski. How's it going, mate? Talking about secondary metabolites, trying to increase them into the plant. You know how healthy they are. You want more? We all want more. Want the stank and dank. Bring on that health. Bring on that health. So I'll get back into which one was it? Actually, which one do I want? Oh, there it is. What happened? Oh. Hang on, I'm lost now. Press the wrong button, sorry. Present, share, screen. Righto. Sorry about that, getting into it. Allow. What's this one? Terpenes. So that was under the terpenes. So let's get into some volatile charts. So what temperature unlocks which cannabinoid and terpene? How about we start with the cannabinoids first? So this is a chart with Celsius and Fahrenheit for all you F-heads in the US. It's not F-head, you know, that was Jesus. I'm sorry. Fahrenheit schedule, and this is the Celsius schedule. I hope you're laughing now, you know I'm joking. So I'm just reading the chart. So at 120 Celsius, the THC drops its acid molecule and makes it go into its reactive THC compound. And same with CBDA, that happens at 130, and CBC is 140. So these are the temperatures on the first chart, Fahrenheit's in the second one, and the different cannabinoids in the third one, and the compounds and different things that are inside of those. And so as you go down here, you can see that the temperature increases. So if you're going to go and smoke your medical cannabis at 120, you're probably not going to get the high or the potential of the molecules that you could get. So if you were to put the temperature up to 160, you'll find that, cool, you're going to go and get a different type of effect. You're going to get the delta 9 out of the THC, but you won't get the delta 8 out of the THC. So there's different effects that are in this. So then if you were to put it up a bit higher again to 190, you'll find that you're going to unlock your delta 8, your delta 9, and your THC A out of that. So it's going to give you the full effect. So that's how to work THC. And then the CBD has the same similar profile, just in different temperature ratings. It's all to do with temperature. 130 to 266 F is where you drop the acid molecule to make it come into its reactive form. And then if you want to make CBD active and be volatiles, so it's might have a little, it's essential oil type thing. And then it's got this little round oil bit. So to reduce or to make that, get the best out of that and shrink it or make it gone so it gets induced into you, it's got to get up to these temperatures. and they're just different other cannabinoids, CBN, CBE, and this chart goes in and shows you it's at benzene's unlocked. Benzene's a really bad hydrocarbon, and it causes you harm. So if you're going to smoke your cannabis above 225 Celsius or about 450 Fahrenheit, you've got a high chance that it's going to combust, and that combustion is going to unlock benzene and other hydrocarbons, which are really not good for you. I've got a volatile organic compound meter, and it sets off if I was to do it in my vaporizer at 230 Celsius. If I go and like mortise and pestle it up, all the after vape material, which I used to do, and then smoke at 230, it makes a big dark cloud, and it really affects you a lot, but it's not healthy. So I stopped doing it, and it used to give me bad wheezing, a bit extra wheezing. And then after I found out about the temperatures, and I went, oh, that's why. So it's trying to be healthy. Well, don't try to do whatever you want, but this is the best way that I promote. Now what's a cannabinoid? No way. Oh, the cannabinoid family tree while we're here. So it starts off as CBGA. That's like you could call it mother. Then as it goes on a bit, it naturally turns into THCA, which is good for different properties, anti-cancer, anti-inflammatory, anti-spasmodic. And then it turns also into CBDA, CBCA, and then the separate one, again, it'll form CBG in lower amounts. And then once you heat it up to those temperatures I was saying before, just above those, you'll end up dropping the acid molecule off, and it'll change into its reactive form, which has other different anti-properties as well. And you see, once it's turned into this form, you can see, look at the different amount of anti-properties there are. Actually, I shouldn't do that if you want to compare. There's a lot more. So it pays to know the temperatures to unlock them at. That's why I really like my digital volcano or the digital mighty. So you can get the best out of it, so you know you're exactly getting what you want. And then after it's aged, it turns into CBN and CBN. Oh, there are two there. Oh, because CBD ages into CBN, and THC also ages into CBNA. Oh, is there a CBNA to unlock at? CBNA? No. That would have been interesting, because we can see it must be high. Because after it's aged, it takes off the heady amount, and it gives it a more of a sleepier effect, the CBN, when it changes after you smoke it. And that's talking about something that's stored for as low as 6 months. And if you want to store it in a high temperature, it could be as low as 2 or 3 months. You've really knocked out this, taken off the 20%, for instance, and increased this 1%. What am I showing you? I want to show this chart in terpenes now. I'll stop sharing that. Any questions going through? Hello. It's good. I see everybody's chatting. Oh, here we go. What's Kemet say? They use eugenol for mucus removing during corona. So that's one of the eugenol. So is that a mono or sesca? Not quite sure, can't remember. But yeah, you use one of those terpenes for mucus removal. So there you go, there's a good example as well. Thanks, Kemet. Okay, is that for vape? No, this is, put a question mark at the end, can you, mate? And then I'll recognize it a lot easier. Is it for, no, these temperatures are in the oven. These are the temperatures anywhere. With your lighter, within a joint, within any device that you consume it, even in your body after it's consumed. If you even eat raw edibles, it can only get up to your body temperature, 37 or 100 F. So that's what will be unlocked. So it's anywhere. So this is how it functions. So to get the best out of your medicine, you want to make sure in any type of form that you're consuming it, that it gets above these temperatures for you to unlock those properties beneath it. I'll get some now of some terpene temperatures so we can see some of those, because that was a pretty good explanation of the No explanation. That's a good cannabinoids done. Where are we going here? All right, might just have to start and look. Let's start and look. I haven't prepared this very good. No. Yes. Here we go. So, this is a study done comparing the dates, different technologies to see what happens with different ones. So they kind of tested the same and see what's come out. So I'm only interested in the latest. So it's this right-hand side here. So I'm only going to discuss that because the left has changed. As science ages, it changes. So beta-caryophyllene, which is very good. That's the spicy one. You might sneeze, and it's also good for pain. And that's temperatures is 260 Fahrenheit or about 130 Celsius. So these start relatively high-ish. So you have to, so if you're going to smoke or vaporize it below these, temperature beneath 110 Celsius, you can see you're not going to get anything. Or 250 Fahrenheit, you're not going to unlock any of these terpenes. These are just examples. And if you want to get some myrcene, which is the pain, solid, a good pain one, 167. So if you're thinking, oh, I really need the pain, well, you must vape over these temperatures. What's another good example? Little terpenol out of, that's this terpene you get out of turpentine. You'll know that one. Excuse me. That'll do it. There's nothing real. Oh, some up here. No. Pinene, humulene. All right, I'm just going to squeeze through, try and get into some terpene benefits. No, I want temperatures, thank you. There's some rad pathways there. Yeah. Allelopathy, we were talking about that before. I want some more temperatures, please. This is a really good, I suppose I can talk about this at the end. This is 55 pages on every terpene, and or not every, but a lot of terpenes and their secondary metabolites related to medical cannabis and what they do, their medical effects. So we'll just go into one, cannabidiol, all right, CBD. No, what's myrcene, the pain one. Anti-inflammatory, oh no, I can't read it. Nociceptive. Oh, what a bad example. Well, oh, that ended pretty bad, didn't it? Might be talking about them then. I can't remember what I was up to. Oh, that's right. I was looking for the actual charts. I remember now. Sorry about that. More temperatures. Here we go. So humulene is very low, just above boiling point, just about 107 Celsius or 225 F. And that's some of its good properties in there. All this, anti that. That's why medical cannabis used properly is so medicinal. Look at all these anti-immune protectors, blockers, inhibitors, sedatives, helping this, helping that, it's just excellent. And for me, having epilepsy, antispasmodics, it's just so effective. So back to the temperatures. So these are just a rough idea. Caryophyllene is very common, myrcene, eucalyptol, you know the eucalyptus smell, lemon, limonene, little like lavender, terpenol, as in terpene. So these are all different things. So see, if you wanted to try and get terpenol, you're going to have to smoke it very, very high, or vaping at a high amount, which is nearly at the level where we're going to unlock all those bad hydrocarbons. Already been through that. Terpenes and CBD oil. Well, that's because they must support CBD oil, but the terpenes that in medical, just a terpene is a terpene. Isabidol, woo, floral. And properties, antimicrobial, anti-irritant, anti-inflammatory. That's right. Phytol, yes, that's the go. Let's try to get more temperatures. Terpene health benefits, yes. There's heaps. So if you want to go and secondary metabolites, yeah, this is actually it. If pain relief, it's very common. So you'll go down here and you'll go look at H, So you want some alpha-humulene, you want some camphene, you want some eucalyptol, some alpha-bisabidol, and B, just name a few, and boronanol. Why isn't myrcene there? Oh, maybe I'm wrong then. Where's myrcene then? I'm really sure myrcene. There, brain functions. Okay. Well, I'm real sure it's with pain relief, just because it's not in this chart. Doesn't mean to say it's not. But this is an example of how you can also use all these to your health advantage. So if you want to sedate, if you have problems sleeping, you might want linalool, or one dominant in terpenol. So you'll have to get that terpenol up to the high temperature of 219 Celsius to unlock that. And a linalool, you can check that temperature and you get that up to that, and that'll be your sleeping tablet as an example. Guide, here's some more terpene temperatures. Vaporizers at. So if you want a high linalool strain, you'll probably want to try some kosher kush, some Romulian, or some sour kush. They have a dominant linalool terpene. And then after that, you'll want to hope that your post-harvest techniques from the grower have been maintained so that you'll have this high in your end result. Because they all test 2 or 3 weeks after they've done a harvest. And so that's the numbers that you get. So after it ages, oxygen is horrible for breaking things down, as you know, and it's stored everywhere. Even in cryovac or shrink wrap bags, there's still oxygen inside of it, and things break down. So they change. So especially after a few months. In Australia's medical cannabis that's imported, there's a very low, mega low chance of these being in here. Mega low chance, because there's involved with the smell. If you open your bag and it smells not much, it's very weak, it's lost a lot of its terpene and flavonoid profiles. The tail matters, yes. This was to do with how they're synthetically chopping the tail in the lab, and then by reducing the tail, it changes its molecule, so that's how they synthetically can change CBD into THC. And it's been already banned in 3 states in the US. So stay away from synthetic cannabinoids. Government trying again to try and not win, but do it their way. Leave it up to the plant. I like this little chart because it does show a lot of things. So I'll start down the bottom here in Fahrenheit, but it just shows the different types of temperatures that are unlocked with different types of effects. It just goes up, just gets higher and higher. I'm not going to explain them all. Been through a fair bit. Achieving the entourage effect. That's the mixture of the cannabinoids with your terpenes and flavonoids. So the mixture of those two together gives a different outcome. So if you've got very high dominant in terpenes, if it's terpene profile, say 5%, which is really high, that means you've smoked it around that time that it hasn't aged enough, and it's been post-harvest operations have been preserved to it, so you still have 5%. It would test, say, 2 or 3 months afterwards. That's going to give you a different effect, entourage effect, to if it had a very lower terpene percentage effect. Because out of that, you're probably getting just a higher cannabinoid effect, because these don't really shrink that much. They just they change a bit, they age, but the terpenes lose. They're volatile and they just evaporate and go. It's sad. Cannabis vaping temperatures. This is, a vapor person would have wrote this, that's why it says it. But these are the temperatures that just unlock it. So it's not just to do with vaping. This is just the temperatures which unlock it. So see, look how low. CBG down here, 52. I said it was 60 before. And OC mean is 60. It's not that high. So they're 2 of your lowest ones that are volatile at that temperature. So if you want to do an extraction at 100 Celsius or 212 Fahrenheit, you're going to lose all of these terpenes. They're all gone. They're just volatile. They've changed their form. They've been heated up. So that means they've turned into their reactive form and they're just into the air. Bye-bye. Look, see ya. Is that the last one? It is. Whoa, this is good. So there's heaps of temperatures. What else is there to discuss? I said I was going to wrap up with something. Hey, it's been an hour. This has been pretty good. And I'll just say good day. I saw Dave there. How you going, Dave? Dave, what's up? Does the temperature when you decarb your cannabis change the cannabis when you decarb at different temperatures? Yes, because you'll be unlocking different. Here we go. Let's explain with a chart. This one. So if you, what was the question again? Just to accurately answer it. Does the temperature when you decarb your cannabis change the cannabis when you decarb at different temperatures? Yes, because it'll be decarb. Well, back to this one. If you decarb it at a low temperature down here, it won't change the cannabinoids. So I suppose it's really to do with this 140 and 280 F. So if you were to decarb it really high temperature at 140, see how there's an actual range here in between. So if you were to fine tune THCA, it ranges in between 60 and 125 Celsius or 140 F, 257 F. So of this, I'd try and change, I'd go the lower end of it. And then you can tell by the difference when you smoke it. So you can do your own experiments on what your molecules will react at different temperatures, because you have to consider the terpene and flavonoid profile. So if you're going to go and decarb at 140 up here, you're going to lose all of those ocimene, CBG, and any other terpene that is below 140. The humulene, I think that was really low as well. That was just over the boiling point, remember? Beta humulene. So you're going to go and lose all of them. So it makes a massive difference, mate. Great question, thank you. There's one above it too, sorry, Kemet. Can that 160, 190 temp be reached in a joint? Oh yeah, in a joint. Get a, to test that, you'll get one of those temperature guns, and as you suck in, you'll see the red cherry come up. Put the temperature gun on the red cherry and you'll see it'll get up to about 400 or 500 Fahrenheit. It'll get right up there, mate. It combusts. That's that process called. It's where it produces, that's why you get cancer causing things out of it, because it goes above that level where the benzene and the hydrocarbons are unlocked. Let me show you. See down here, they said it's at 205 or Celsius or 401 Fahrenheit for the benzene and hydrocarbons. I don't believe that, because my volatile organic compound meter, it reacts about a bit hotter than that. When it's at about 228 to 230 Celsius, which is probably 450 Fahrenheit. Hope that helps, Kemet. Let me know, Kemet and Dave, if it's anything to add on that. Does anyone ever notice how people who don't partake in the herb are less healthy and usually afflicted by some ailment or disease? Says Lene. Yes, that's an interesting statement. It's true. And once people get onto it, they start to get all of these benefits from different types of terpenes, flavonoids, and the other health properties that are in medicinal cannabis. If it's grown and harvested in a healthy way. There's a lot of people who, this is another whole talk nearly on the harvesting procedures that people do where they lock in all of the unwanted chemicals. So the plant needs 17 essential elements for it to grow, and it can harbor and lock in all of those to be at a high amount upon harvest. So you're smoking it, you're getting a different head high, your throat's sore. I don't like it at all. So, yeah, it makes a big difference. Yeah, facts. That's right. I'm sure. Renee says, I've sure noticed that cannabis consumers are healthier and tend to live longer, happy lives for the most part. Exactly right. Yeah. Interesting that, isn't it? They're much more mellow and everything sort of, as long as they're on it. But it's a true statement. Having all it's health, to do with health. And the doctor, they'll say, why do you want this cannabis? Well, and you've smoked it for 20 years. You say, what? He says, or she says, what health problems do you have? Well, you don't have any health problems. You just know it agrees with your body, and that's why you consume it. So there'd be heaps of underlying areas. If you didn't consume it, I'd have more stress, I'd add more this, I'd add more that. So it's just an overall health booster. I see it. I love it. I'm an eyes open till eyes closed, 2 to 2.5 pounds a year smoker. I love it. Right. I'll see if there's anything else. Oh, yeah, look, there is. I've got a Word document down here, which was a good conclusion. These are some plant tissue is a continuous and reliable source of secondary metabolites that prevent overexploitation of plant from its natural habitat. Plant secondary metabolites come in small quantities but play a major role in plant defenses, as I've previously explained. In addition, they are used for pigments, dyes, insecticides, cosmetics, nutraceuticals, pharmaceuticals, food additives, flavors, fragrances, all sorts. So they're so beneficial. So not just for us to consume, they're also good for the environment, for our daily uses of things. Advantages of using plant cell. This is to do with tissue culture, because tissue culture is one of the best ways to get secondary metabolites, because it's in such small amounts. You just do tissue cultures of those cells that you want to grow, and then you'll choose the ones that you want that have the highest profiles, and then you'll go and exploit those. So this is just a paper on that. The major advantages of using cell cultures for secondary metabolite production include plant cells are carried out in controlled environments. Oh, okay, let's get to something. Why did I put this up? There's a good reason here. Disadvantages. I haven't even said any advantages yet. Strategies for increasing secondary metabolite production. Oh yeah, I remember now. So some strategies to increase secondary metabolite production in your plant cells in your cannabis plants. So how can you do that? Well, I've spoken already about activating the pathway, jasmonic acid or the octadecanoid pathway. There's where the innate defenses and the MAMPs can be operated to, microbial assisted molecular patterns. There's a few here that gets into it, about cloning right and growth medium, but a cool ones are precursors. So you can go and give a precursor, and that's, what's an example of a precursor? I hope this has it in it. It doesn't. Well, how can I say that? I wonder if it's got an example of a precursor. I'm just trying to, I know them, but I'm trying to explain them for you. I'll just read what this says. It involves the exogenous, the exterior supply of compounds which are intermediate or apply at the beginning of a biosynthetic route of the desired secondary metabolite by aiding in the culture medium. Okay. What that means is right at the end of the pathway, I'll just use this as an example. So right at the end of the pathway, you'd go and add some dihydroflavins, and then the plant will recognize that, and it's only got to do one step to get the end result. That's the precursor. So it's before the step, before the cursor keeps continuing, it's the pre bit before it. I hope that explained well enough. Where was I? None of these look familiar. Oh, gee whiz. This one, was it? No. Oh, the Word document. So precursor. Yes, an example of good precursors are, I actually can't tell you my secrets. An elicitor is another form of stimulation for secondary metabolites. An elicitor, they're signals that trigger the formation of the secondary metabolite pathways. So they would be something like it's getting an attack. So you got the FLS2 gene would be, no, the FLS2 receptor would be activated if bacteria with a flagella were there. So the bacteria would be like an elicitor, you could call it. So if you were to go and, with the bacteria were there, or you to put them there, or they were to become there, that would be the signal that would trigger the formation of the secondary metabolite. And that changes differently. So remember, there's in the MAMS, the microbial activated molecular patterns. Oh, actually, it's just up here. Into this section, there's different receptors and pathways activated from different approaches. Like if you've got your umycetes, like Jono has over in Thailand at the moment, his PEP13 receptors will be activated in his medical cannabis plants. So he wants to somehow turn that off. So you can breed that in, mate. I've got fungal resistant genetics. Yes, very handy. That's where some of this stuff was getting into, and we're not going into that today. It's pretty full on. That was an example of that. That's right, Word document. So that's an example of the elicitors, just different ways to activate the secondary metabolite profile in your plants. Because it's all about stimulating, or it's all about the secondary metabolite profile and the cannabinoids. That's the whole reason that we grow mostly medical cannabis, to get those properties out of it. So if you can activate them or help the plant in aiding to do so, even if it's not ready to do so, you're at a very, very good advantage that you're going to get the genetic potential out of your genetics. So this is a cool conclusion, that plant cells can be considered as the potential source for production of important plant secondary metabolites. Since the production of secondary metabolites by plant cells is dependent on environmental conditions and quality fluctuations, fast-growing and high metabolite producing cell lines can be established by adopting certain strategies like selection of parent plants, supplying precursors, application of elicitors, and by special treatments such as biotransformations and immobilizations. Thus, the application of these strategies can help in exploiting the genetic potential of your medical cannabis plants as a source of secondary metabolites. Cool. Cool. Yeah. All right. Anything going on? Does anybody? Oh, I mentioned that. Does anyone ever notice? Oh, yeah, facts. That's right. Reading the paper. Reading the paper. Yep. I've been going for an hour. That was good. Thank you for the suggestions last week for putting today's show on, for the people who suggested that. That little button down the bottom, the dollar button, is for appreciation, and for subscription, the subscribers are welcome to subscribe. There's 4 little classifications under that. One's a thank you each month. One's $80 for some lectures that I haven't released in my playlists. $100 is for someone who wants a one-off call going through medical problems and they want my opinion. And $300 is more so for a company or someone starting out and doing legals. I only support legal activities. So that's for the subscriptions. And under my playlists, I don't mention this much, but I've got heaps of free lectures. So from all of my studies, I'll save a lot of the private lectures and they'll go under my playlists. So if you look under there, there's thousands and thousands of hours of university lectures that show the slides of the topics that I talk about. That's how I learn. So, yeah, hit them whenever you want. I don't earn nothing off them either. They're just to help, because I'm here just to help. I virtually don't make any money out of this. Well, I don't really. It's unfortunate. One day, though, that's why I keep trying. One day, you got to try. Yes. Well, it was a great topic today. Is there anyone got any questions on that? I know yeah, it's a great topic. Yes. Thank you. One of my faves. Yeah. Well, it's to do, especially those temperatures. If you know the temperatures, you can get the best out of your medical cannabis. It's really rad. And then to use your microscope afterwards in your after vape material. So if you save that, use a microscope, see if you've got any round bulbous trichomes. They'll look sessile. They won't have any stalk capitate on them. So they'll just be round and just sitting on the brown leaf or green leaf material. And if you still see them, that means they've still got properties inside of them. They haven't actually shrunk and gone down to nothing. So you can extract or use them somehow. Yeah, it's a good way to tell if you're using all of your plant material. And for those in Canada, where I used to live, or North America, they're allowed to do that. And that's a good way, after you're doing your extraction processes, is to see if everything's done. Like if you want to do some rosin, you'll put it under your heat press and you'll press it, and then you'll see, oh, have I got rid of it? You check your cake that you've made, and you'll find, wow, look at all these trichomes in there. Well, yeah, that's still some good stuff that you can go through another extraction and release them until so the little microscope helps. You don't need much, just a little microscope. That's a good secondary metabolite profile tool you could use, because that way you can see how the cannabinoids are grown, at stage they're at. Did the grower grow into when they're a little bit early? Did he grow into where he let them finish late when they're a little bit amber? Because there's different effects that happen in them. And another good myth too about the amber trichomes looking CBN. Well, if they've got CBN in them, which some people say that's what they think it is when you age them, aging means a longer period or a higher temperature. So get a test result. They'll see on their test result sheets of their amber trichomes that there's very low CBN. So that'll tell them, or that'll explain that it's not quite aged, and the THC hasn't quite aged enough yet for them for the biosynthesis for it to change its pathways. So that's that one myth that somebody brought up about the trikes. I suppose I could show more. Oh no, I've been over an hour. I need to show any rad trikes. But yeah, talk about that. And next week will probably be an open topic. So I'll just be answering questions again. So yeah, if you've got any good questions, save them up till then even. And then I'll try and show some slides. I should get back into a pattern soon. I've been able to have the news and have all the latest information about that, like I have previously done in the first 6 months of the show. I'll have nearly been going for a year in a few weeks' time. Not bad. And I thank everybody for helping. Just being there and just asking questions helps a lot, because otherwise I'll be talking to myself, and that's really not good. So I appreciate everybody for trying as well. Like I do, it is hard. It's not easy doing this. I dedicate a few hours of my time each week to try and help others through education with medical cannabis. It's all that's needed, that's for sure. Yeah. All right, well, there's not any questions rolling through. I reckon I can do what Ned Kelly said and show some peace and say hooroo. Right, well, thanks everybody for turning up. I really appreciate it again. Thank you, everybody. Very nice words. Appreciate your support. I'll see you all this time next week. Have a happy week and happy breeding, happy growing, and good health to everybody. Everybody. Bye-bye.